hummingbird, obviously, right? And what do hummingbirds eat? Sugar, right? I mean, they're like birds on crack. They're very hyper. That's all they do is drink nectar, right? They drink nectar. Now, they eat insects. I don't know if you know that or not. They do eat some insects. But they nectar mostly off of flowers or off of feeders, right? Everybody feeds them now. This is a Costa Rican hummingbird. So, gosh, go to Costa Rica and there are lots of hummingbirds, right? There's not just one, like here in the Midwest. So, they're fantastic. There are about 80 species of hummingbirds, but they all eat nectar. This one is a beautiful one here. But think about it. So if I eat nectar, it's all sucrose, so a disaccharide. And basically that sucrose ends up in the intestines. And so now the big question is, well, how do we get it into the blood supply, right? And so that's kind of where we're going to go. We're going to start talking about how membranes, cellular membranes do this, right? So we got to move sucrose, which is a charged molecule, and quite big, really, honestly, right? From the lumen of the intestine into the bloodstream. And all the blood vessels are underneath the epithelium, right? That lines the intestines. So it's trapped, right? It's trapped in that body compartment, and it's the epithelium's job to move it from one side to the other using transcellular transport. And so that's what we're going to start talking about, is how cells do this. All right, so here's kind of where we're moving from like chapter 1 and 2 to now to chapter 5 in that textbook. These are kind of the page numbers from the 4th edition. And we're going to cover these things right over here. And so probably one of the hardest and most confusing early topics in biology is osmosis, right? Osmolarity. You guys remember struggling with that ever? Now I'm going to really make it hard. So it's not too bad though. Once you figured it out. So I don't know how much you've talked about osmosis before, but there are differences between osmosis and tonicity. Right? Those are two different kind of properties of the different body compartments, and we'll talk about them. We're going to spend a lot of time talking about transport and diffusion and how we move things. So here is a beautiful epithelium, right? This is a simple columnar epithelium, like the digestive tract. You can see they're really tall cells. And so notice here we have apical membrane up here. How do I know that's the apex? Because you can see those little microvilli, right? So this would be, for example, the lumen of the small intestine, right? Down here is the basement membrane. It's not lit up real well. You can't see it really well, but see how all the cells kind of end? All right, this is connective tissue down here. All right, this is all connective tissue. See all these fibers? It's all collagen. All right, it's a collagen. And these are the nuclei. Notice they kind of all line up. So down underneath the nuclei, this would be the basal compartment. Above the nuclei would be the apical compartment. All right, this is a specialized cell type right here called a goblet cell. So these are all cells that are involved with digestion, right? This epithelium. So that's why I chose it, because we have the hummingbird there in the beginning. So, reminder, I told you this the first day. Physiology, everything happens in a membrane. 99.9% of physiology happens in a membrane. Membranes are important. We need to remember this, right? That's why I spent the last week doing it. All right, so body compartments are obviously important. These fluids we're going to be talking about are important. We talked about homeostasis and which fluid homeostasis is controlled in, and we call it the interstitial fluid, right? Or the internal environment, like we talked about in the first lecture. Now, just to give you an idea, this is an old figure, but it gives you kind of an idea of any mammal that's about the size of a typical man has about 28 liters of intracellular fluid overall. Your billions of cells, right? And about half of that, another half amount of fluid, about 14 liters, that's plasma and interstitial fluid. Notice most texts don't even talk about lymph. Lymph is a very small contributor to the extracellular fluid. Notice that about 25% of plasma, or of extracellular fluid is plasma, and about 75% is interstitial fluid and lymph. They kind of treat them as the same thing. This is your internal environment, right? the interstitial fluid. This is where we maintain homeostasis. All right, so this also gives you kind of a more schematic view of our fluid compartments. All of those are separated by membranes, right? By cell membranes or anatomical membranes. Now, what this shows you here is just a schematic drawing of a blood vessel here. So in this blood vessel that's coming down is the smallest blood vessel in the body. They're called capillaries. Most of the blood vessels in your body are capillaries, right? You have 60,000 kilometers of blood vessels running through a typical human body. Think about that. Almost all of that is capillary, right? Or capillaries. So capillaries are the blood vessel that deliver materials to the tissues of the body. So any tissue you section through is going to have capillaries in it, right? To give you an idea, in a mammal like you, most cells are no further away from a capillary than 2 to 3 cells. And you're billions of cells. That's why capillaries are so common, right? Because most cells are receiving their nutrients, their oxygen from a capillary. They're dumping their waste products, their carbon dioxide, back into that capillary, which sweeps the blood away and delivers it back to the heart. Now, plasma is what those red blood cells, that's what these represent, are traveling through, right? They're being swept around in a fluid called plasma, which we know is about 25% of the extracellular fluid. But guess what? These fluids are linked to each other because plasma can give rise to interstitial fluid. How? Simple. It leaks from the capillaries into the interstitial space. So you can see this capillary in particular right here, where these 3 cells are found, have little holes in their epithelium, and plasma can leak to form interstitial fluid, right? What's really interesting are the lymph vessels. This is another set of vessels outside of blood vessels, follow the veins, which are one of the blood vessels of the body, down into the tissues to form lymph capillaries. And the lymph capillaries collect extra interstitial fluid and then deliver it back to the heart. So you have fluid in some tissues leaving the blood, entering the interstitial space, and then being collected by lymph vessels, which carry it back to circulation and then dump it again. So your interstitial fluid is circulated just like plasma. It's not stagnant. It just doesn't sit there. It is actually circulated. This keeps the interstitial fluid fresh so that these cells, which are sitting in the interstitial fluid, can receive everything that they need, right? And also get rid of anything they don't need, and they live happily ever after, right? As long as things are going in perfectly within the tissues. So these are how tissues are typically set up. Now, the thing is, we got to move things in and out of these cells. We got to move things in and out of the blood. We got to move things out of the cell into the interstitial space, and from the interstitial space into the cell. That's where the physiology happens, right? Where all the nitty-gritty happens. The other thing that we have to do is we got to keep the solute concentrations in each of these fluids the same, so that water is not moving dramatically in one direction or the other, right? Because water tends to want to move and diffuse just like solute, and water can pass through a membrane. So water can move from one compartment to the next with no help, right? And it can do that, all right? So what I mean by that is, think about this. If all of a sudden the cells have a lot more solute in them than the interstitial fluid, then water is going to move into the cell, and the cells are going to swell. What happens when a balloon swells too much? Bursts. That's not good. You start blowing all your cells up, right? That doesn't hold well for your body or that tissue. So we got to keep osmolarity the same in all of these fluids while we're moving a bunch of stuff around. I mean, that's hard. It's hard to do, right? It's hard to understand how we can do that. And so that's kind of where we're going once we start learning kind of how all these cells do that. And it can start to become complicated and kind of overwhelming. So We got cells are bathed in fluids, and all physiology really happens in water. So that's where our chemistry happens, right? So most of the biochemistry that happens in physiology involves water in its reactions anyway. So we have three different major fluids here. We have plasma, interstitial fluid, and intracellular fluid. I know we mentioned lymph. Lymph is going to be part of the interstitial fluid, right? So they just kind of clump it together, even though they're in different places. So what we're going to start talking about is how cells and then body compartments that are lined with epithelium can basically have a completely different osmolarity, first and foremost, than their environment. When I say osmolarity, we're talking about solute per whatever our solvent is, right? The concentration of solute in our solvent. Our solvent here is always water, right? And our solutes are going to vary, right? So anything that can dissolve in water is going to have a charge to it. So ions are the big ones, right? Ions are the big ones that we are going to deal with. We have lots of different types of cations, which are positive, and anions, which are negative. And what's really interesting is when physiologists started looking at the differences between the interstitial, and plasma, they see a major difference between what cells concentrate on the interior versus what's found in the interstitial fluid. And so the reason that cells can do this is because of the proteins they express. All right? And so they can start moving these anions, cations, and other solutes around to concentrate them on one side of the membrane or the other. While doing that, they keep the osmolarity the same on either side. Okay? And if you just stop for a minute and think about that's phenomenal, right? You're moving ions at different rates and at different concentrations, but you're maintaining solute concentration equality or equilibrium across the different fluids. If that breaks down in animals, it's a big deal. It causes massive damage or destruction as far as physiology goes. Also, we know that sometimes body compartments are going to increase in a solute that we need to move. And to do that, we're going to use epithelia. That's one of epithelia's major physiological jobs, is to move things from one body compartment to the other. We used the example of the hummingbird yesterday, right? Where they eat nectar, all that sugar enters the digestive tract. Now we have to get it into the blood. And so it's the lining of the intestines that do that. So we're going to start talking about some of the incredible things that vertebrates can do, like fish. Fish are strict regulators of their solute concentration. This shows you pond water. All right, so the fish are completely amazing, particularly freshwater fish. So are the saltwater fish, because they maintain so much higher a solute concentration than the fresh water. All right, and so you can imagine that they have to deal with water movement a lot, particularly the gain of water, right? Because water is going to want to flood their interstitial fluids, flood their cytoplasms, because look at the solute concentration difference. Right? If you just look at sodium, for example, it's 160 millimolar, and it's 0.5 millimolar, right? So they're going to want to gain water. Water's going to want to move in, right? Because the concentration's so much higher here. And also the salt is going to leave, doesn't it? It's going to want to leave the fish and try to dilute their body fluids. And so they have to have evolved a number of mechanisms that allow them to maintain their salt concentration and deal with that water intake. And that's what we're going to do next as we move into doing some physiology of some organ systems. We're going to do kidneys and gills and watch how they work. Now, if you look at this, I like this graph down here. This shows you intracellular fluid versus interstitial fluid versus plasma. And what you notice, if you look at the interstitial fluid and the plasma, they're pretty similar, right? And I told you yesterday that the plasma gives rise to interstitial fluid, which is collected as lymph and returned. So you're circulating plasma and interstitial fluid. So you can see they're really similar as far as what they look like. But when you compare them to the extracellular fluids, to the intracellular fluids, it's much different, right? And we're going to talk about how we do that. So, cells are in osmotic equilibrium, but chemical and electrical disequilibrium. So, I want you to think about that. Good test question, right? Explain how cells are in osmotic equilibrium, but chemical and electrical disequilibrium. And that's what we're going to start talking about right now. So osmotic equilibrium simply means that the solute concentration on one side is equal to the solute concentration on the other side, right? But what cells solutes decide to concentrate are going to be different than the solutes that are found in the extracellular fluid. So it simply means equal numbers on either side, but different solutes, right, on either side. So this is kind of how we're going to do this, and we're going to use pumps and other proteins to do this. And so this is probably, in my opinion, one of the most important slides of the whole semester. Right? It's what every cell it's ever been looked at in a vertebrate body does. It maintains what we're going to call a resting membrane potential. And this resting membrane potential is a measure of charge. Right? It's a measure of electrical charge. Now, I just said that cells are electrically in disequilibrium. So what that means is the charge difference in the extracellular space is going to be different than the charge difference on the cytoplasm. And so you might wonder, where's this charge occurring? It's occurring at the membrane. No surprise, right? All right, it's occurring at the membrane. The membrane is a wonderful insulator, electric insulator. It's hydrophobic, it's lipid, it's neutral, right? It doesn't have a charge, and it acts very much like an electric insulator. And what we're going to do is we're going to have a difference in charge across the membrane. So what I mean by that is on the surface of the extracellular leaflet, it's going to have a slightly different charge than the surface of the intracellular leaflet. The charge occurs all at the membrane. What's incredible is if you stick an electrode that measures electrical current into the extracellular fluid, it's neutral. If you stick an electrode into the cytoplasm, it's neutral. If you get close to the membrane, you start getting a charge difference. It's going to be slightly positive on the outside and slightly negative on the inside. So if you put an electrode really close to the outside of the membrane, you'll get a positive reading. If you stick the electrode through the cell membrane into the cytoplasm, you'll get a slightly negative reading. And we're reading in what we call millivolts, so really small differences in electricity. Now, believe it or not, and I'm going to teach you this later, one of the coolest things, have you guys heard of electric eels? They can produce electricity. The large Amazonian electric eel can kill a person if it distributes enough charge towards you. Stop your heart. It can produce like 300 volts of electricity. So that's like sticking your finger in an electric socket, right? And they can do that by producing a bunch of cells producing electrical current and then focusing it, a discharge of it. Now, this is a difference in electricity, right? Positive versus negative. So how do we do that? All right, how do cells do that? Every cell has done this. Every cell they've stuck an electrode into, muscle cell, nerve cell, pancreatic cell, intestinal cell, skin cell. It doesn't matter what cell that if you put an electrode into it, it measures negative charge. So they all use the same exact mechanism to maintain this charge difference. And that's what this figure represents. All right? So what I want you to do is I want you to listen to me, make sure you understand this, and I want you to be able to draw it out and explain it because it's a test question on the first exam. And damn, if you get it wrong, I'm going to be mad. I'm going to start doing shots of tequila. All right? Because I just told you it's on there. All right? Matter of fact, if you all don't get it right on the first exam, I'll put it on the damn second exam until you get it right.
I thought you were
well, if y'all get it right, we'll take shots of tequila. All right, how's that? Don't tell anybody. Probably get fired for that. Right? And now I have a puppy I got to feed, so we can't get fired. All right, so how does this work? So all cells have a pump, all right? The best studied pump of all ATP-derived pumps in all cells, and there are several of them, is the sodium-potassium pump. All right, so the sodium-potassium pump has it's called an wow, that marker doesn't work. Hopefully this one does. It's called an electrogenic pump. So electrogenic versus electroneutral. So neutral versus genic. What that means is an electrogenic pump aids in maintaining a difference in concentration gradient, that is, a difference in charge. So what does that mean? You gotta pump ion more on one side than you bring in, or vice versa. So with the sodium-potassium pump, we pump 3 sodiums out to 2 potassiums in. So we're pumping more positive charge out than we're bringing in, right? And so what that means is this is going to aid the effectiveness of 2 channels that are called leakage channels. All cells have these 2 leakage channels, potassium leakage channels and sodium leakage channels. They leak 24 hours a day, 7 days a week, your entire life. All right, it's like having a boat that's leaking water all the time. All right, and then what the pump does is gather the water and dump it out, right? All right, and so notice, we're going to maintain, if we have sodium-potassium pumps pumping sodium out and potassium in, then what's going to happen? You're going to build a huge sodium gradient out here and build a huge potassium gradient in here, right? Sodium's moving in, lots of sodium in the cell, less sodium on the outside of the cell. Lots of potassium in the cell, little potassium in the outside of the cell, right? So you got huge concentrations of sodium and potassium. But notice we're pumping more sodium out than we're bringing in, right? As far as potassium goes. Also notice the 2 leakage channels differences. These 2 arrows mean something. There are a lot more potassium leakage channels than there are sodium. So in cells that are very electric, have very large resting membrane potentials, nerves and muscle, there are 30 or 40 potassium leakage channels to every 1 sodium. They give you kind of a number difference, right? And so you're leaking a lot more potassium back into the extracellular fluid than you are bringing sodium in. This difference is what causes my charge across the membrane, because a lot more positive charge is leaving than is coming in, right? This is only happening at the membrane, this charge difference. All right, you're like leaking and you're pumping, right? And it's just happening right there. And so it gives you this slight positive and slight negative leaflets, intracellular versus extracellular. So when you start pumping stuff and forming concentration gradients, that's a form of potential energy. And in this instance, it's also a form of electrical energy. This is a definition of a battery. All cell membranes are batteries. So think about a Duracell battery. You stick in something, right? I mean, I don't know what nobody uses batteries anymore. Joysticks, my kids' joysticks is the only place they put batteries in anymore. So all a battery is a positive end and a negative end separated by an insulator, right? That has some fluid in it. And when you touch or connect the ends, electrons move, right? And it powers our devices or our accessories. The same thing here. We have a nice insulated barrier, the membrane, and then we got a slightly positive and a slightly negative end. If you connect the ends, you're going to get electrical current, right? That is, for example, if we have a bunch of sodium out here, right? If I put a sodium channel in here over here, don't worry about the leakage channels because they're always there, right? If I have a gated sodium channel over here and I open it, sodium will move into the cell, right? And you'll be able to measure that because it's an ion. It's an ion that's moving. Remember, all electricity is everybody had some physics? All electricity is a movement of a charged particle. In physics, it's electrons, right? In biology, it's ions. So when ions move, you can measure that. You can see that change. So when you connect the outside to the inside using channels and you're moving ions, you'll go from a negative 70 millivolts, you open the sodium channel, to positive 30 millivolts because all that positive charge flows in. So this is a form of what we call potential energy, and this is how cells store energy. It's the most effective way ever built to store energy. Now you guys remember your freshman biology class, right? You remember general chemistry. You know the law of thermodynamics. It's really hard to capture energy, isn't it? Because no matter how you burn it or whatnot, right? Most of that energy escapes as heat. It's impossible to capture heat, right? It's really hard to catch heat. And so when cells burn glucose and make ATP, all right, that is pretty ineffective. So the best way to use the ATP that we do make is to build concentration gradients. And so cells do this no matter what type of cell they are so that they can take advantage of that potential energy because it's endless. These pumps are working all the time. And so you always have this bit of potential energy that I can use to do work for the cell. So nerves and muscles, they automatically expend about 30, I've seen anywhere from 25 to 40% of their ATP stocks every day on maintaining concentration differences. All right, so that they can run muscle and nerve physiology. 40% of the energy that they make is already invested in these pumps. So all cells invest a tremendous amount of energy to running pumps. These pumps are essential, all right, in cell physiology, but they're also essential in osmolarity, all right, which we'll discuss in a little while. So it's these leakage channels that really run the positive and negative, and it's the pumps that help maintain these concentration differences so that the leakage channels can work, right? So think about this. If the pump shuts down, these are going to continue to leak, right? And they're going to continue to leak and continue to leak until they reach what? Equilibrium, right? That is, eventually enough sodium flows in that they equalize. Enough potassium leaves, they equalize, right? Now, I will tell you this, though. In an animal, the potassium leakage is not that big of a deal. You lose some potassium, you might lose some water with it, maybe a little bit. But really, it's sodium leakage that is dangerous. The more sodium that leaks, the more concentrated your cytoplasm gets. Eventually, water starts following it, and cells swell, and then they explode, right? I mean, they'll blow up eventually. So without the pumps, you can get massive lysis across your body. So these pumps are there to maintain the concentration differences. It's these leakage channels that cause this. Now, this negative charge on all cells is called resting membrane potential, all right? Resting membrane potential. All cells have negative resting membrane potential. And on the test, I'll ask you how. What's the mechanism? And you're going to draw me this and explain it in clear detail, and I'm going to celebrate, right? So look over here at this picture here. So this is what a typical cell looks like. So the major anion in the cytoplasm are proteins. Proteins and phosphates. Those are kind of your major anions. And if you think about it, that makes sense. Most of the proteins we build stay in the cell, right, where they're utilized by the cell. And a lot of proteins have a lot of negative groups on them. The carboxyl group's negative. A lot of the amino acids are negative. Phosphates are also negative. I mean, we have some free phosphates. It depends on the cell. But most phosphates are involved with ATP, right? Or they're used by kinases, but there are a few of those. The major cation in the cytoplasm or cytosol is potassium because of the pump, right? In the extracellular fluid, the two major ions are sodium and chloride. All right, sodium chloride, right? That's where most of your salt. So chlorides are not permeable, sodium is not permeable. We pump sodium out, we keep sodium high out here and low in the cell. We keep potassium high in the cell and low in the extracellular fluid. Another ion that's really critical that we are going to talk a ton about is calcium. All right, calcium is another really important ion, and there is absolutely almost no calcium in the cell. All right, so calcium is strictly regulated. Most calcium is either found extracellularly or it's stored in an organelle, the smooth endoplasmic reticulum. All right, so the smooth ER is a calcium storage site for cells. And you might ask, well, why do we keep calcium low in the cell? Calcium is a major second messenger. All right, and so remember, second messengers change physiology in response to a ligand, correct? And so we don't want just calcium sitting around in the cytoplasm because it'll turn on enzymes we don't want on, right? So we got to keep it low until we need calcium. Okay, everybody good with this? Again, I keep saying everything is layered. If this, I really need you to go home and memorize the hell out of it. I want you to draw it, explain it to each other. Get a whiteboard. They have whiteboards here. You can draw. Make sure you know this because we're going to build on it, right? We're going to build on it as we go through the semester. So, as I mentioned, nerves take advantage of this. So this is a figure out of an animal phys text that shows you this is a nerve up here. Hopefully you might recognize this. If you haven't had any nerve anatomy, up here this is the cell body of the nerve. And this is the long process of interest here that's called the axon, right? The axon is a wire. I mean, it really behaves like a wire. It has the ability to conduct electricity just like your wires that power our computers and our iPads. But we're moving a different ion. We're conducting a different ion. I shouldn't say ion. We're conducting a different charged particle. We're moving ions instead of electrons. Now, if I blow this up, just this little box, that's what this is showing. This is showing you in yellow. This is the axon, right? And then you got extracellular fluid on either side. And notice that nerves maintain very strict negative charge on the inside, very strict positive charge just on the outside. And look at the sodium and potassium differences, right? Just like I mentioned before. So sodium's really high outside the cell, very low in the axoplasm or the cytoplasm of the axon. Potassium's very high on the inside of the axoplasm and very low, right? They're pumps all along this membrane and leakage channels that are maintaining this resting membrane potential. This form of potential energy using charged particles is called an electrochemical gradient. An electrochemical gradient. So this is one place. One place you may have also heard that term before, at least I use it when I used to teach it, is the electron transport chain. This is exactly what the electron transport chain does in the mitochondria, right? It pumps hydrogens into the intermembrane space there between crista, and if you pump a bunch of protons in one direction, which are positive, it gives you a charge difference across the membrane, right? And then we use that charge difference to make ATP. Here, we're going to use resting membrane potential, this electrochemical gradient, to do work for all cells. All right, all cells are going to use it a little differently. And we're going to talk about that as we start moving things from one side of the membrane to the next. Does that all make sense? Yeah. So we call this voltage, all right? Resting membrane potential is a voltage difference across the membrane. Now, as I mentioned, just to show you, again, a really old figure here from one of the original tech. I think this is actually out of the first PowerPoint slide I ever made when I taught like 25 years ago. So it's pretty old. It gives me lots of nostalgia. But in any case, I said all cells do this. They maintain these differences and across vertebrates. So this is a squid nerve, a crab leg nerve, frog sartorius muscle. And when you look at them, there might be some slight differences in the numbers, right? But still, sodium's high on the outside, low on the inside. Potassium's high on the inside, low on the outside. It doesn't matter what cell you look at and what animal, whether it's invertebrate or vertebrate, the concentrations are the same because this mechanism is old, right? Evolutionarily, obviously, right? Helps us regulate very different ion concentrations from our environments. Okay, so love these kinds of figures, as I've already mentioned. So let's use this electrogenic pump and leakage channels, this resting membrane potential to do work for the cells. So, energy requirements of a membrane. If we want to do membrane transport, we can do it in two different ways, right? We can do passive transport and we can do active transport. All right, so what's the difference? If you remember, passive transport uses concentration gradients to do work. That is, they just use concentration gradients and allow those gradients or allow that solute to flow down its concentration gradient. That's the term I use, down. So you're always moving from high to low, right? That requires no energy. It's just what we call diffusion, right? Is the term that we use. So it uses the energy that is in the molecule, and they call it molecular motion. All molecules are moving. If you concentrate them in one area, they tend to want to move to areas of low concentration. It doesn't require any energy from the cell, right? Doesn't require anything from the cell. We just use the concentration to do diffusion. Now, there are two types of diffusion when you involve a membrane, all right? So the big difference between diffusion in chemistry and in biology is we put a membrane in the way, right? We insert a membrane into this now. And so if something's hydrophobic or really little, really small, you can do what's called simple diffusion, where it just crosses the membrane, right? Just goes from one side of the membrane to the other with no help. And that's called simple diffusion. So, for example, oxygen. When we breathe oxygen in, we bring it into our lungs, it just crosses the membranes of the epithelium in the lung tissue into the blood. Why? Because oxygen's really little, and it's also non-charged, right? It's not hydrophobic. I mean, it's not hydrophilic, sorry. And so it just passes right into the bloodstream because it can move through simple diffusion. Now, the hummingbird example. The hummingbird's nectars, all of that sugar ends up in the intestine, dissolved in the fluids of the intestine. So how do I move it? Now something bigger has charge to it through to the blood. Now I'd have to do facilitated diffusion. I still have a concentration gradient, don't I? Bunches of sugar in the intestines, very little of it in the blood. So the sugar wants to move, right, from the intestines into the blood, but it can't because it can't cross the membrane. So what is the intestinal cells going to insert? They're going to insert a what into their membranes to move the sugar? A sugar channel, right, or transporter. And then that's called facilitated diffusion because I need help, right? I need to insert a protein in order to do it, all right? That's called facilitated diffusion. So these are the two major types of diffusion you see. Now, I will talk real quickly about osmosis, which is diffusion of water, right? Water will always move down its concentration gradient, all right? So cells, remember, are going to do a really good job of keeping the concentrations of solute in the extracellular fluid the same as far as number in the intracellular fluid. So water doesn't move directionally. But I'm going to teach you that sometimes the body wants to move water from one body cavity to the next. Right? And so it's going to want to move water into the trachea, for example. And we're going to talk about how the body does that. So that is basically moving materials without using any cellular energy. The other form is called active transport, right? Which is on the yellow side. So this is passive transport in the white. This is active transport in the yellow. Active transport requires ATP either directly or uses ATP indirectly, right? So, how do we use ATP directly? Using ATP directly means I actually take the AT, break it down, add the phosphate energy directly to the protein of interest if I'm using a protein. So, primary active transports or pumps, PP, right? Primary active transport or pumps. Pumps actually split ATP directly and use the energy to move something across the membrane against its concentration gradient. So what pumps do are pump things out from low to high, okay, not from high to low. So they're pumping materials, ions, other solutes from a low concentration to a high. We're continuing to increase the concentration on one side of the membrane. So primary active transport uses ATP directly to do this, to build concentration gradients. So pumps build concentration gradients. Now, once I got this huge concentration gradient on either the inside or the outside of the membrane, then I can do secondary active transport, which is going to use the concentration gradient to move things against the concentration gradient of another solute. All right, so we call this mediated transport across the membrane using proteins we're going to call SIM and antiporters. And they are going to use a concentration gradient to pump things across the membrane. And you might ask, and this was always a question I ask, is why don't we just have pumps for everything, right? Why don't we have a glucose pump, a hydrogen pump, a potassium pump, a sodium pump? Because if you, first and foremost, pumps require ATP, and we just don't produce enough ATP energy to run every type of pump that you can think of. But a concentration gradient is much more effective than individual pumps. So we use one pump, the sodium potassium pump, to run a bunch of proteins that can use concentration gradients. It's much more effective. The body's about 35% effective in use of ATP energy. A lot of it still escapes as heat, but we have come nowhere close to making a machine that effective, like that burns gasoline, for example, or uses coal energy or whatever it is. So the body is really effective in capturing, at least partially, the energy from ATP in these concentration gradients. And really, you can store that indefinitely. It's not like you're going to lose it, right? I mean, as long as the membrane's intact, you can make a concentration gradient and use it immediately, use it 2 days from now. It doesn't matter. The gradient's always there. So secondary active transport is using ATP indirectly by using the concentration gradients to pump stuff, okay? I'll give you some examples here in a minute. The other way we need ATP is direct use is through endo, exo, and phagocytosis. So these involve the membrane in moving large amounts of material via vesicles. All right, so we're going to use the membrane to pull stuff into the cell or to release stuff out of the cell. That requires the use of the actin belt that I mentioned yesterday and using motor proteins like myosin. And we know myosin uses ATP, right? So if I want to move the membrane around, I have to use ATP energy. So all vesicular transport is active transport. Okay, so far so good? Okay, so start talking about water, and let's get into osmosis, which can be confusing. So we know a membrane is a nice phospholipid hydrophobic barrier. Things that are little and uncharged can move easily. Small, uncharged polar molecules, now they have to be really small, okay? Like water, glycerol. Another one we'll use is called urea. These are charged, but they're partially charged. Everybody remembers the difference between partial charge and full-on charge, right? So ions, you either lose an electron or gain some electrons, right? The charge is real. It's a negative, positive. They attract each other. Partial charge is when you got covalent bonds and one of the atoms has a larger electronegativity. It's got a bigger nucleus. It draws electrons to it more often than the other atom. Oxygen, hydrogen. Big oxygen, hydrogen, small. The electrons, remember, flow around both, right? Because, I don't know, I always tried to envision that their electron orbitals merge, right? And so you're moving around both of them. But as the electrons travel around the oxygen, they stay there longer, right? And then go around the hydrogen really fast. Because of that, often the oxygen is slightly negative and the hydrogen is slightly positive, right? That's what partial charge is. And so things like water, which have partial charge, glycerol, urea, anything that's really little that has some partial charge can bang its way through or pinball its way through the phospholipid layer. And so it's slower. It takes a second for it to work its way through, but it can if there's a big enough concentration gradient. These guys that are hydrophobic and uncharged, they fly across. No problem at all. Now, larger polar molecules, glucose, sucrose, amino acids, right? They're big and partially charged. They can't cross the membrane. They're just too big and too charged, right? Anything that's an ion or that has true charge, like positive and negative, anions, cations, cannot move across the membrane without help. Now, water, I already mentioned, it can move. It's slow, but it will move if there's a concentration difference. Everybody knows solvent versus solute, right? So diffusion. Diffusion is driven by concentration gradient mostly. And so there's a mathematician that came up with what's called Fick's law or Fick's diffusion equilibrium. This is what it looks like. There are many versions of it. Don't worry, you don't have to memorize it. You don't have to use it. I'm not that type of animal physiologist, even though my damn animal physiologist class made me use it. All right, but this is the simplest form of this. J represents diffusion. And then what you have here is rate equals surface area, right, times concentration gradient. So these two here, the P and the A, they represent surface area. And then you have to have a concentration difference, right? If you don't have a concentration difference, then you don't really have diffusion, do you? Now, do ions and water and things still move across the? Yes, they do. They just move equally, right? In and out, they're in equilibrium. There's no directional move. But if you do have a concentration difference between 1 and 2, then you can actually get a positive rate, right? A rate of movement. Surface area is really important. The higher the surface area, the higher the rate of diffusion. The lower the surface area, the lower the rate of diffusion. There's a direct relationship between those two. And what you're going to notice is that cells, if they really want to move stuff, are going to increase their surface area, right? Microvilli are a great example of this. Cells that move materials in hollow organs often have microvilli because it increases surface area, which increases diffusion rate. Now, we're taking advantage of just the random movements of ions. So when you concentrate them together, they bounce off of each other, they move. Things like increasing temperature means that diffusion's faster because the ions are moving faster or the solute is moving faster, right? So for example, your high body temperature means diffusion's pretty effective in us. I mean, your core fluids are 98.6 degrees. That's warm, and so diffusion works really nicely. If you got a lizard sitting outside right now and frozen, it's not, this diffusion rate's going to be really slow, right? Because the fluids are really cold. And so that's one of the advantages of being a mammal, right? We can have high diffusion rate because of our warm body temperature. Now, there's a video down here. I'm not going to click on it. I'll probably throw it into the lecture video. Did you guys see I've been putting lecture videos up? I think some people are watching. I'll throw the video in here so that you can see it, but it gives you an animation. So, great visual representation of this. Here's a membrane. Here is one side of our membrane has lots of solute, the other side does not. And if we're permeable to that blue circle, it's obviously going to move relatively quickly, right? From one side to the other. Now, flux does happen in the other direction. Flux is movement from one side to the other. Flux, it goes in both directions. This is very directional, right? So the net flux is going to be from the high concentration to the low concentration. But this is what a pump would do. It would pump, for example, these were sodium ions. This is what the outside of the cell would look like. This is what the inside of the cell would look like, right? Building that huge concentration gradient. Now, we can change or modify Fick's equation to include thickness of our barrier. This is where it becomes biological. So that equation I showed you a minute ago is for like physical chemistry, like, a solute concentration in a solution. When we add a membrane, then that adds a barrier. And so we can change the equation to look like this. All right, well, rate is going to be surface area, concentration 1 and 2, over x. X is the width or thickness of the barrier. And this can be seen in a couple ways in animals. All right, so for example, the first way is, if we're talking about epithelium, the taller the epithelium, the bigger the x, right? X is just, how thick or wide something is. So, for example, simple columnar epithelium is much taller than simple squamous epithelium, right? So columnar epithelium has a much larger x in this equation, a number, if I wanted to give it a number, than squamous does. The larger the x, the slower the rate, right? Because we're dividing by it, right? And so, the larger the x gets, the slower the rate of diffusion. The smaller the x is, the faster the rate of diffusion here. Just using basic math. So, if I want to move oxygen really quickly in the lungs, what do you think is lining the lungs? A simple squamous epithelium or a simple columnar epithelium? Simple squamous, right? Exactly. Now, the other place where this becomes really important in animals is we have this animal cell here, and we want to move the green dots out, right? And so if we start at 100 millimolar here and 0 millimolar here, and let's say that this green circle is permeable, it will start to move into the extracellular fluid, right? As it moves into the extracellular fluid, you can imagine it's going to start building up right outside the cell, right? As it moves from one side to the other. Takes a while for it to fuse out. And so your concentration gradient is going to go up. It's going to go from 0 to 2 to 3 to 5 to 12. And so what's going to happen to the rate of diffusion over time? If this boundary layer here gets bigger and bigger, is it going to speed up or slow down? It's going to slow down, right? As these numbers get closer to each other, diffusion rate slows, doesn't it? Right? So, one way cells can deal with this, because this becomes inefficient. If it gets into the 20s or 30 millimolar, which it would, it gets inefficient. Things might move too slowly. So how do I deal with this? Well, why don't I just sweep this away? Right? Why don't I just move it? If I move it, and then I'll keep it near 0, won't I? So one way that life started dealing with extracellular fluids is what we call circulation. We start moving stuff, right? So if I want to keep diffusion rate high from one body cavity to another, I will remove the boundary layer by producing flow, all right? And so this type of flow is called bulk flow, when I start sweeping the extracellular fluid away so that I can continually move something at the most effective rate. Does that make sense? And so we don't want fluids stagnant in the body. We don't want to, especially the extracellular fluids. We don't want them just sitting there. We want to move them or get some circulation going so that we can prevent the boundary layer from forming when we're moving materials from one body cavity to another. And we can keep it low, and that will give us effective diffusion. Make sense? So flow rate's very important. Even if it's slow, even if it's really slow flow rate, it's going to be effective because we can sweep that boundary layer away.